So if we look at these pieces that make up the inner ear, we have the cochlea. The cochlea's claim to fame is what it really houses into it, and it houses the receptors for hearing. The receptors for hearing are found within only the cochlear duct. The receptors for hearing are only found within the cochlear duct. And as you look at this thing over here in cross-section, right? Okay, here's the cochlea in cross-section. We have a lot of stuff there. Okay, and the cochlear duct is right there. Okay, in the middle. That's the only spot where I have the receptor cells. Everything else has other stuff around it. I'll talk about that on the next slide. The vestibule, this region right here. Okay, this region right here. Oops. Okay. Contains two important structures. Well, three, but we're going to keep it two for now. Something called the utricle and the saccula. These house receptors for static equilibrium. These are going to tell you if you are standing still, right side up, upside down, moving up an escalator, moving up an elevator. Okay, it's going to be these two guys, the utricle and the saccula, found only within the vestibule, that are going to be helping with our ability to determine static equilibrium. Semicircular canals, there are three of them for the X, Y, and the Z axis. This is dynamic movement. Okay. It will house receptors at the ends of all of those semicircular canals or semicircular ducts to allow us to interpret that we are moving to the left, to the right, forward, at an angle. Okay, that's what this does. Okay, so we got static and dynamic motion here. So if we look at that cochlea here in cross section, it actually houses three different ducts. Okay. This book calls them the cochlear duct, and I'm going to give you some other names as well for these guys because it depends on the author in the book what they're called. Okay. The cochlear duct is also called the scala media. Okay. And here you can see it, the cochlear duct, scala media. M of media, M of middle. The cochlear duct or scala media is in the middle of the three ducts. Okay. So here is my cochlear duct right there. Okay. There's my cochlear duct. The other two are easy to figure out which is which when you know where the middle is. So here's my middle. And in the middle, you can see it houses it's kind of bound by this vestibular membrane up top and a basilar membrane down on the bottom. Okay. And it houses the most important thing here, guys, and that's the spiral organ. That's going to be the dude that it's picking up sound. It's also called the organ of Corti as well. And we'll talk about that on the next slide. But we have two other ducts. We have one that's here underneath. Okay. We have one that's underneath. Okay. That's the scala tympani or scala tympanum, singular. It is adjacent to the basilar. So what does that mean? It's below the scala media. For me, I visualized, or this is what I told myself, okay? I tamp things down into the ground. I tamp things down into the ground. Scala tympanum was always below scala media, or the cochlear duct. The scala vestibule, you look up into the vestibule at church, right? To pray to the gods. Scala vestibule is above scala media. Okay. You go into the vestibule to look up at church. You tamp things down into the ground. Below, above, in the middle. Okay. There is also this thing called the helicotrema. The scala vestibule and the scala tympani right at that little spot right there are actually connected. And that's the helicotrema. Okay. That's where the upper chamber and the lower chamber are connected to one another. So as sound waves are traveling through the scala vestibule, if they reach all the way to the apex, to that center axis of the cochlea, they'll enter into the scala tympanum as well. And sound waves will dissipate out that duct. Scala vestibule, above. Scala media, in the middle. Scala tympanum, below. Perilymph, perilymph, endolymph. Okay. That cochlear duct is membranous. The other ones are formed by the vestibular membrane, the basilar membrane, and the bony area of the cochlea. So that's the bony labyrinth. If we get up close and personal here with that scala media, here is what the spiral organ, or organ of Corti, but we'll use spiral organ because that's what this one uses. Okay. The spiral organ is the sensory structure for hearing, is sensory for hearing. Okay. So when you pick up the sounds coming out of my mouth into your ears, this is where the magic is happening. Okay. This is where that magic is happening. Okay. It's within the cochlear duct. We have hair cells. Okay. And here they are. Okay. There's an inner hair cell. There's an outer hair cell. Hair cells. Okay. We're not going to get into the semantics of these guys. All right. If you want to know, it's the inner hair cells that are actually providing the bulk majority of our ability to hear sound. And when I say bulk majority, like 95% of sound is picked up by these inner hair cells. Okay. As you can see, they have cilia extending off the heads of these hair cells. Okay. They are referred to as stereocilia. Stereo means what? Sound, right? So these are going to be the dudes involved in our ability to pick up that sound. Along with along with a handful of stereocilia, we also have one very long hair and we refer to that as the kinocilium. Okay. So there's the long one. There's the kinocilium right there. And then all the other guys here are stereocilia. They're just not up there blowing in the breeze. They're actually embedded in, so you can kind of see it right here. That little goo. That little goo is called the tectoral membrane. Okay. So those cells are embedded in that tectoral membrane. Right? If those hair cells were not in that gelatinous membrane, remember this is a fluid-filled chamber, right? And what would happen? Okay. Those would be like seaweed at the bottom of the ocean, right? Just moving with the tides, moving with the current. So every time you moved your dang head, you'd move the fluid in the ear, and that would move those hair cells, and that would cause a depolarization event. Good God Almighty, your ears would be ringing all the time. So they are embedded in that tectorial membrane to keep them anchored until a sound wave causes the depression of fluids within these various different chambers, which is going to move the tectorial membrane, and magic. And we'll see that here in a second. Okay. Spiral organ houses the receptors for hearing, the inner hair cells with their stereocilia and kinocilium. The spiral organ is found within this middle chamber, this middle duct, called the cochlear duct. And this cochlear duct is filled with endolymph. Surrounding it, above and below, scala vestibule, scala tympanum, respectively, filled with perilymph. Okay. Basilar membrane on the bottom, vestibular membrane up top. That's what we have right now. Okay. So again, if I was to ask, give me sound waves moving from outside of the environment into the cochlea to be interpreted, sound waves get funneled into the ear via the pinna or the auricle, travel through the external auditory meatus, vibrate the tympanic membrane. As soon as they vibrate the tympanic membrane, that moves the malleus, the incus, and the stapes. As the stapes oscillates over that oval window, the fluid within that chamber, which chamber? Scala vestibule, will start to move through it. Okay. Okay. So that oval window opens up into this right here. So as the fluid gets displaced, as that foot plate of the stapes moves over it, okay, that'll cause that little flap of membranous tissue to actually get sucked back this way and then come back, and that pushes the fluid. So the fluid gets displaced within the scala media, or I'm sorry, within the scala vestibule here. As it gets displaced in here, what does it push down on? Pushes down on that vestibular membrane. Okay. So now what in the hell happens? Okay. If I'm pushing down here on this vestibular membrane, what's that going to cause to the fluid within the endolymph? To be depressed down, right? And now what's it going to push down on? The tectoral membrane. Okay. What's embedded in that tectoral membrane? all of those stereocilia. Okay. So we get pressure waves now within the endolymph of the cochlear duct. All right. Hair cells get distorted. We get neurotransmitter release. Travels through the cochlear branch of the vestibulocochlear nerve, cranial nerve 8. Sound waves travel through the scala vestibule, then start to slowly dissipate, and then they travel through the scala tympani, where they now exit out the round window, and you stop interpreting the sound. Let's see what a picture of all that nonsense looks like. Okay. So here's the sound waves traveling through the external auditory meatus. It's that tympanic membrane. Moves the malleus, the incus, and the stapes, and the stapes oscillates over that oval window, causes the wave of fluid to move here, and you can kind of see it eventually dissipates, pressing down on that tectoral membrane, pushing down on those hair cells. And the wave, because this pushes down here, right? Which pushes down here, and this little flap of basilar membrane pushes down, right? And so that's how this little wave of fluid movement can now occur within the scala tympanum. And it dissipates out the round window as excess sound waves are lost. This right here, the sound wave could go right here and stop. Okay. This sound wave can go all the way to here and stop. Okay. It depends on the frequency. So that's how sound is moving through this thing. Okay. Sound waves have been changed into mechanical energy. Where did that change occur? At the eardrum. Change that sound wave into now mechanical energy. I'm moving stuff. I'm moving that connective tissue membrane, which causes the movement of the malleus, which is connected to it. And you can actually see it when you look into an otoscope. Moves the incus. Moves the stapes. Stapes moves the fluid within the scala vestibule. And now that fluid movement starts to travel. Okay. How far that travels in the cochlea depends on the extent of movement of the stapes, how loud that stimulus was. Depressing the vestibular membrane, pressing down on the tectoral membrane, hitting those hair cells, sound, eventually causing the wave to depress the fluid within the scala tympanum, and it dissipates out through the round window. So let's see what actual mechanical changes are happening in that cochlear duct, okay? Because that's how the sound waves move, all right? Scala vestibule, depress stuff, depress all the stuff within the cochlear duct, and then fluid moves in the tympanum and excess sound waves, bye-bye. But what is actually happening in that cochlear duct? Okay. Here's what it looks like. Okay. Here's my hair cell. Here are my stereocilia and my long kinocilium. As that sound wave travels through that scala vestibule, it presses down on that tectoral membrane. As it does that, it causes those hair cells to move. Okay, there's that inner hair cell. Okay. As that is pressing down, okay, it's coming down like that. It's a wave that's coming down. It's coming down. So as that comes down there on top of those little hair cells, causes those things to move. Okay. And my fingers are all kind of weird. Okay. But it causes those things to move. See how my fingers are moving? That is basically what is happening as that wave of movement of fluid pushes down to the scale of vestibular fluid, pushes down that vestibular membrane, pushes down on the endolymph, pushes down on the tectoral membrane, which pushes down on those hair cells, on those stereocilia that are coming up from those hair cells, right? That are embedded in that sucker. Okay. Those hair cells, you could put inner, outer, I don't care 2 bits. Okay. I don't honestly care at all. Okay. 95% is inner, so you use it. All right. I want you to kind of understand the general gist. Those hair cells contain ion channels. Remember, this is the endolymph. The endolymph is all potassium. Potassium moves in. Potassium is the one that causes depolarization. It's the only one that does. Okay. So here we have what are called protein links, tips. These are just protein fibers at the tips of the stereocilia. That's it. Okay. That name is insanely stupid, right? Tip links. Good Lord. Whatever. Okay. It's just a protein fiber at the end. If you look up really close, that's what it looks like. Okay. Just a string of amino acids from one tip to the next. That's all. All right. But those tips, those tip links, those protein fibers are connected to an ion channel. It's potassium. So these tip links, when they get bent, okay, when they get bent towards the tall one, okay, that tall one actually pulls all those tip links and opens up all of those potassium ion channels. Potassium comes rushing in. Okay. And this causes a depolarization. So as soon as that little wave of depolarization moves through the scala vestibule and eventually comes down and reaches that last point where it just pushes down on that vestibular membrane, which pushes down on the tectoral membrane, which pushes down on those hair cells. As soon as it does that, boom, potassium rushes in, depolarization. Okay. As soon as that wave went down into that endolymph, it pushed down on that tectoral membrane. What else does it push down on? That basilar membrane. The basilar membrane moves, right? Something's sitting on top of it, it's moving everything. So sound waves now enter into that scala tympani. And it starts to move out. As soon as that sound waves start to go back in the opposite direction, what happens to that wave of fluid movement? It exerts a force back up. So it pushes up on that basilar membrane. So now where do those stereocilia and kinocilium? They come back to where they're supposed to be, right? And what happens to the pull of those tip links? Don't happen. So if nobody's tugging on those tip links to open as they bend back, now that causes all of those ion channels to close. The wave of depolarization stops traveling down the cochlear branch of the vestibulocochlear nerve. And now you don't hear anything. Have you ever been to a loud rock concert? Probably. I hope so. Well, before life stopped. But if you don't wear ear protection, when you walk away from the concert, you hear that constant ringing in your ears. It's because that huge amount of loud sound caused the tectoral membrane to be smash down on those little hair cells. So even though there's no sound waves coming in the ear, those hair cells are still bent. They're still open to a degree. Those tip links are still pulling it open. So you hear that ringing in your ear. That's why you put those earplugs in. Okay? It lessens the sound that's pushing against the eardrum, that's moving the malleus stapes, that's moving the fluid within the scala vestibule, that's pressing down on the endolymph, which presses down the tectorial membrane, which presses down on those hair cells. And if you're exposed to those constant loud sounds, you can damage those little stereocilia. You can bend them, break them. And that's going to cause a constant influx of potassium, which causes a depolarization, which you are going to hear as the ringing in your ears. Pretty cool. Pretty cool. Pitch is just dependent upon the frequency of the vibrating. Okay. High pitch, low pitch. Okay. You can see kind of high frequency is caught in the first part, the initial entry into the cochlea. The basilar membrane, is very, 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 very stiff. Okay. So it takes a very high frequency with amplitudes that are very, very close to one another, which is what a high frequency sound wave would be. As the sound waves get lower and lower and lower frequency, they can travel further within the cochlea, as you can see from the picture there. And so those are going to travel very, very longer towards that helicotrema. So the low frequency is caught by more of the inner parts of the cochlea itself. So when they test for your ear function, your ability to hear sounds, that's why they test both high and low frequencies to make sure that the integrity of the entire cochlea itself is intact and you can hear sounds. The last thing here when it comes to sound waves is loudness. And loudness just depends on the wave amplitude, the degree to which the sound waves are entering to the ear. If it's really loud, you have a huge number of waves coming in at a very short time frame. So that's going to create a much more louder sound for your perception. If it's a very smaller sound, then those waves don't have the huge amount of amplitude coming in. And we interpret that as something that's very, very quiet. Where does it all go to? I mean, it's all going to the primary auditory cortex, right, in the temporal lobe. That's where we are ultimately heading to. But it extends from the cochlear branch of those hair cells, right? Cranial nerve 8. They enter into the medulla oblongata, and the medulla oblongata has a cochlear. Medulla oblongata has some cochlear type nuclei right there. And you can see it extends to really the first major thing from the cochlear branch of the vestibulocochlear nerve to the inferior colliculus. Okay, the inferior colliculus was involved with reflexes, with sound, right? It also goes to the superior the olivary nucleus, and that's going to help initiate those reflexive actions of the muscles. I didn't go into superior olivary nucleus, so I'm not going to expect you to know that one. From the inferior colliculus, we're going to go ultimately to the thalamus. In the thalamus, we have a number of various different nuclei there that allow us to figure out in what direction. Is it coming at me? Is it going away from me? So the thalamus is going to put together a lot of the pieces of information to allow you to figure out if you need to get the hell out of the way, or if you need to go to the sound to figure out what it is. And then from the thalamus there, we go to the primary auditory cortex, which you all know as the temporal lobe itself. And that's where we consciously perceive, oh, I hear the train coming at me and I got to get off the train tracks. Oh, I don't know if I'm going to ask it. I may incorporate it into a question or something. There are a couple different types of deafness. There's what's called a conductive deafness and a central neural deafness. Now, look at the words. Look at the words. Okay, so now you're looking at the words. All right, so conductive. Conductive just means the wave is being conducted from the outside towards the cochlea. All right, so with conductive deafness, it's just an interference with the wave. This could be you got too much earwax, you have something stuck in your ear canal, you have otitis media, you have old age, where you have some arthritic changes occurring with the auditory ossicles, all right? Something that interferes with how the wave is getting from the outside towards the inside, okay, towards the cochlea. Central neural is actually the inner ear problem, okay? So this is a problem with the cochlea. This is a problem with the spiral organ, okay? This could be a problem with the actual hair cells, stereocilia, or unfortunately the cranial nerve itself, okay? And it's easy to figure out. You do a Weber-Renée test, and it's pretty fun to figure out. Not fun for the person, but fun for you to do.